But the questions that are raised by the electrical engineers and the questions that are raised by the physics community with respect to signal-to-noise ratio are things that I think fundamentally need to be answered if we're going to wind up with mechanisms that lead us to understanding what the potential biological effects are. Because I think it's reasonable to propose if the biology can't detect the signal exists, it's not going to respond to it. All right. Now, one of the techniques that we've looked at is we said neural networks are a popular thing in computer science as a way of doing pattern recognition. They're derived in the computer science world from basically looking at the biological systems. One of the questions I think worth asking is let's turn that around. Say let's use what we know about neural networks from the computers and say how do we use that kind of information to decide what a biological system can detect. And we've done some of that at 60 cycles by looking at saying what's the lowest signal at 60 cycles that we can get a computer network to detect as a function of the input signal to noise ratio. And we found by running some simulations that if you started out with a signal to noise ratio of 1:1, it took about 100 runs to train the neural network to recognize that 60 cycles was there. But the way you trained the signal was important. You had to tell it, for example, 59 hertz was not 60 in order to do that. The interesting thing to me was that if we turned the signal to noise ratio down so that the signal was 0.01, it took only 1,400 runs to train the network to recognize 60 cycles with 97% accuracy. So there are adaptive processes that we need to understand. Okay. Here's an example of one of the things that's worth knowing. This is some experiments that Howard Wachtell and I did. It's a pacemaker cell on the top of an aplysia. This is old work. If we apply a microwave pulse to it, it turns out it slows it down. And in fact is, if we wind up providing a big pulse, and I'm talking about microsecond pulses of kilowatts, what happens is we shut it down. Or what I've shown here is it slows down. It turns out that it's not only a function of temperature. As Carpenter showed, if you raise the temperature of this pace speeds up, and here we're slowing it down. It turns out the rate of change of temperature is important as well as the temperature. We don't understand this in the detail that I think needs to be done in order to look at what's happening here. Here's some high-power pulses. This is some of the first work we did back in the 75s. We applied something like 3 kilovolt signals, microsecond pulses. On the left, you have the normal size of the membrane. On the right, you have clearly damaged membranes where those errors are. On the average, over 6 minutes, this set of pulses is below the threshold of the standards. Right now, those have been modified since 1975. There are peak power limits. But at this time, this was within the standards, and my reaction is, I don't want my membranes and my mitochondria looking like that. So it says, you don't cover everything. You need to know. But it's not an average temperature rise. It's the effect of short, high power, high signals, and that gives you something different than you get from slow raises. The average is not a good way to describe what's going on here. So you need to look at the characteristics of the pulse and the characteristics of your signal, and they can make a difference. Here's some experiments that are currently going on. What we've done, and there's not time to go into it, nor is it appropriate to talk in details about these experiments, but what we have done is we've taken a concentration gradient of a chemoattractant. We extract white blood cells from, well, you've heard about tough professors. My students working on this have to give blood. All right, so students can draw blood. We take a look at the white blood cells, and this is the sort of random motion you get on the slide for a neutrophil meandering around on a microscope slide. When it senses a concentration gradient, if you stake at the starting point there up to point number 15, which is 15 minutes into it, you see that the cell starts to wander up the concentration gradient. And this is the way you wind up getting pus around a cut, whatever, is the white cells migrate to the site of the infection or the site of the break. What happens is when we apply 900 megahertz fields, is at 15, and this is the most dramatic case, it turns off and it goes to right angles. Now the fields that are involved here are the order, my estimate is of volts per meter. One calculation shows a tenth of a volt, another shows 10 volts, and what's needed is because we didn't expect to see anything when we did these experiments, that you need a better microwave setup to get a good design on exactly what the fields are. But if they're the order of volts per meter, these cells move at right angles. Take the field off, and they go back up the concentration gradient. We use the concentration gradient as a way of saying that these are good cells, they're active, so we know they're doing things that they ought to be done. All right? This is the most direct way of showing these things. Most of them take off at an angle and whatnot. But they only work about 70% of the time. If you draw the same student drawing blood from the same students on successive days, we get different answers. And this is disturbing in the sense of the usual physics experiment where you expect if you do the same thing, you get the same thing every day. The problem on my book is that we don't control the initial conditions. That, all right, we don't know what the student had for dinner the night before, we don't know that they're stressed out over an exam, who knows? But we get different results on different days. So this is part of where I think we lack understanding on what's going on. All right. Here is probably some of my strongest evidence that we have effects that are occurring at field levels below those where the thermal is. We plot the velocity of these cells as a function of temperature on the bottom curve. And we've done hundreds or a couple hundred experiments probably. And we plot them with the radio frequency fields on at 900 megahertz. And you notice in the range of around 37 to 39 degrees, they move twice as fast as the other cells. Calculated temperature rise is microdegrees. All right? What's going on? Don't have the foggiest idea. As I say, I'm a slow learner. Yes? Okay. I'm virtually close to done. All right? Significance of pulse and coherence, I think, is an important thing. Repetitive things, if you do those experiments that we did on the aplesia that I showed you earlier, you do that 5 times on 5-minute intervals, you get different results on the 5th pulse than you get on the 1st in terms of the size of the response. So systems are adaptive. We've talked about neural networks. Should point out that stress reactions can be both positive and negative. My way of exaggerating this is to say if you see a tiger coming and you're stressed out and you run faster, that's probably a good thing. On the other hand, if you're overloaded with stress, the next stress can wind up being like the straw that broke the camel's back. So these are things that go in both directions. And there is a difference between a biological effect and a health effect. You can say high blood pressure is bad for you, right? What's the first thing that happens when you exercise? Your blood pressure goes up. But exercise is good for you. So you need to look at both actions and what the background is, and I don't think we understand these things very well. Okay, these things can be both positive and negative. We're looking at, I think one of the most interesting things is using electromagnetic fields for therapy at higher levels. We've seen that you can regrow bones, you can regrow nerve cells in the spinal cord, in terms of what Borgnines has done at Purdue. We're looking at the effect of activating the immune system with electric field signals, and it looks very interesting. I hope by a year from now that we'll be in a good position to talk about some very positive things that can be done in this area. So I think there's exciting things. We saw some results of talking about cancer that people are worried about in Davos, where people irradiated one tumor in a mouse that had 2 tumors on them. Both tumors went away, including the one that was not irradiated. All right, what's happening? And I think these are things that need to be looked at. Whoops. Last, I think it's important to emphasize that we don't live in a risk-free world. We can't prove that cell phones are safe against all those things we haven't thought about. And I would expect that the results are going to be a function of amplitude, frequency, repetition rate, and all of these are going to be important in terms of what goes out if we try to unscramble it. If you take nerve cells, each nerve pulse is about the same height, same width, and the information is in the pattern. I expect that we're going to find that if we're signaling the biological system with RF, that the significance is going to be in the pattern in which we apply those signals. Okay, I'll quit with that. Thank you.